Thank you all. Good evening to everybody. Thank you, Dr. Guswarani, and all the dignitaries present here.
My pleasure, sir.
Yes, and also to Dean, Faculty of Engineering, Mathematics and Natural Sciences, University of Bengaluru, and also Dean, Faculty of Medical and Health Sciences. I think Rector sir is not around, Madam. And Vice Rector, Madam Guswarani is here from Pathetula University, and
Thank
she is
you.
also Associate Professor of Bengaluru University. And I am proud to announce today from Dr. Nath Memorial Foundation, we are arranging our beloved Dr. Mrs. Geeta Talukdar Memorial Lecture, who is our supervisor of PhD, and we are going to listen afterwards from our youngest scholar, Dr. Rinini Dastidar. So without wasting much time, I just want to show our one of the senior most student of Dr. Professor Geeta Talukdar. She cannot join through this what we call through Zoom meeting because for network problems, and so she sent the video of her speech. I am just sharing it.
My beloved teacher Geeta Di. It is my privilege to talk about Dr. Geeta Talukdar. Today, I would like to talk some of my experience while working with Dr. Geeta Talukdar. I joined her lab in the year 1982 and did my PhD in the year 1985. We were like a family, and she always cared for her students, not only for their research work but for their personal problem. She gave freedom to her students while working research, and that helped me to be independent in the scientific research field. She was always ahead of her time, and she was the first person who did cytogenetics for the diagnosis of genetic diseases in our state. Nowadays, the term PCR is well known to all, but we did PCR in the year 1992 when I returned from USA with the training of PCR technique. We started PCR diagnosis of thalassemia, and that work was published in the journal Lancet the year 1997. She had more than 170 publications in different journals, international and national journals. She wrote books, and some of her students are doing research successfully all over the world. She was open to everyone and had smiling personality. I was moved by her personality and ingenuity of the scientific work. I left my permanent college teacher job and came to her lab. I am thankful to God for having the teacher like Dr. Geeta Talukdar, Professor Archana Sharma, and Professor Arun Sharma. Dr. Geeta Talukdar was not only my teacher, moreover, she was my guru. Because teacher takes responsibility for your growth and guru makes you responsible for your growth. Thank you.
Thank you. Dr. Madhusudar De was the senior most scholar, one of the senior most scholar of Dr. Geeta Talukdar, and you have heard from her. She was professor of genetics. She has completed genetics PhD on genetics on breast cancer, Dr. Molusnata De, and she is still working in Ramakrishna Mission Seva Pratishthan, Vivekananda Medical Sciences as consultant geneticist. She handled first time Kolkata. They started the PCR, and we have learned PCR from them. So now I would like to request Dr. Ajanta Haldar, also present here. She is one of the my senior what you call research scholar, and also now professor of genetics and lab in charge of Vivekananda Institute of Medical Sciences, Ramakrishna Mission Pratishthan. So Dr. Ajanta Haldar, please. Ajanta Haldar, Ajanta Di, can unmute yourself.
Can you hear me?
Yes, yes. So
Okay.
I am
Good
just
evening.
sharing the screen, and about Dr. Geeta Talukdar, she will speak.
Good
Yes.
evening.
Good evening.
Today I am proud to say a few words about Dr. Geeta Talukdar, the doyen of human genetics in this part of the country. Dr. Geeta Talukdar was born in July 1936 in Pune, India. She completed her MBBS and MD in 1964 from Delhi University, India. She was awarded PhD in medicine in 1967 from University of Calcutta. She served Vivekananda Institute of Medical Sciences, Ramakrishna Mission Seva Pratishthan, and was dean from 1994 to 2000, and also having the post of senior consultant geneticist till last date. She was recipient of ICMR, CSIR fellowship, and 1973 went for training at Karolinska Institute, Stockholm with WHO grant. She was instrumental in setting up thalassemia counseling and genetic lab in Kolkata, India. She has guided more than 20 PhD scholars. She served as task force on human genetics DBT 1995 to 2000, chairperson of DBT. She also scientific advisory committee member of cabinet, government of India. Thank you.
Thank you, Ajita Di, for your description. Just I want to share, she was, as Dr. Ajanta Haldar told, and also Madhusnata De told, that she is our guide and worked as a very senior position, professor and dean, and also scientific advisory committee, government of India. One thing I must tell, that as a in charge, we always see that she is the pioneer. She reached lab in of before of everybody. We reach always late than her. Even we try, we always jump through the stair. She coming by lift, so we try to go by staircase. That we should reach early and open the door of the lab. And sometimes only happen that we go fast. Always we enter lab, she is sitting on her chair with a cup of tea, reading the news, what you call the articles, etc., or working in the computer. So that way, she is the pioneer in thing, and that is the thing we have to know, that how a person personally can, what you call, influence all the people of the institute. So I would like to request our collaborator. I am from Lincoln University College, is the giving us the platform to use the Zoom facility and also collaborating this event. And I would like to invite Professor Gurswarani to say a few words about this collaboration, because she is instrumental for making collaboration with Indonesia, and she is very helpful to and cheerful also to make this event successful with, and she also make collaboration with Bengkulu University and Patpetulai both.
Okay, thank you,
So,
Doctor. Yeah.
thank you, madam. Please, you please tell about your university and Indonesia something so that everybody will be aware.
Okay, thank you. Okay, thank you for giving me a chance to share, this a little bit about this our collaboration. Good evening, everyone. Good evening, Dr. Sandip. Good evening, Dr. Rinini, and also everyone from India and from Malaysia, and of course from everyone's in Zoom and also on YouTube. From Indonesia, from Bengkulu, and also from Curug, from Patpetulai University. Thank you, Dr. Sandip, for giving us to collaborate for the great event. This is for learning about something about this disease and also this vitamin D from, I think, from the great biochemist from India. So thank you also for the Dr. Rinini. And today, so we collaborate with two universities from Indonesia, from Patpetulai University and also from University of Bengkulu. So for special for University of Bengkulu, this is joined from Faculty of Medicine and Health Science, and also from Faculty of Mathematics and Natural Sciences. I see here there's a lot of from University of Bengkulu participant, and also from Patpetulai University. Here also join with us the Dean of Faculty of Mathematics and Natural Sciences, and also Professor Riwandi from Agriculture Faculty. Also, we have here Vice Dean for Students Affairs from University of Bengkulu, from Faculty of Agriculture, and you see here also the head of Department of Biology, also from University of Bengkulu, and from some our colleges from from University of Bengkulu. Okay, so I think we just delivered our gratitude and also our thanks a lot to collaborate with the Lincoln, and also for the here the Memorial Foundation, Dr. Poder Memorial Foundation. And I think we don't stop just until here. This our collaboration. For next time, we really want to develop this our join our collaboration for the next event. So I think everyone can be fun to learn. I think this really good lesson, good lecture from Dr. Rinini. And so just accept this our greetings, our warmest greeting from Indonesia, especially from Bengkulu and from Curug. Thank you. Enjoy the lecture. Thank you, Dr. Sandip. Thank you, Dr.
Thank
Rinini.
you. Thank you. Thank you, everybody. I just also want to share that Dr. Taluknath Poddar was my grandfather, and we have not seen him. I have not seen. And when my father was in class 10, means SPM standard, then my grandfather expired. So, and he done research in 1937. He published a paper and on the parasites, and with his name, the parasite was named. In 1937, the paper was published, and that paper still is citing in this year, 2019 also, that name of the parasite and other papers, that 4-5 articles that time he published, that articles is still citing in 2019, the Web of Science catching the data. So that type of research minded person was Dr. Taluknath Poddar, and you can see the link I have given, the website link of our foundation. And if you want, I can show you, but I think there is less of time. So I don't want to waste time. We are doing some activity, and also we have started one journal, International Journal of Advancement of Life Science Research. And Professor Kushwarani is one of the editorial board members. She agreed to be with along other international members. So our journal also running, and July issue also published. It is indexed in Google Scholar and also other index body. DOI also have. So that is our foundation. We are also doing some charity works and also organizing events, conference. Last December, we organized one event in Kolkata, international conference, 2017 and then 2019. And now this one webinar we are going to organize few, because now the new normal era, we have changed the means physical contact. We don't have to go anywhere, so we can learn more and we can participate and we can communicate. Like we meet Professor Guswain and all of you, that deans and all these persons, we cannot meet physically before. Now it is very easy that we can get connected. Now we can see that Zoom readings is full of 100 people and YouTube also more than 100 people watching at present. So without wasting much time, and there is also some participant like Miss Uli Feti. She is from the, what you call it, Science School of Health Science, Mandala, Indonesia. She is also present. I will call after the lecture. So now I just, everybody know about Rinini in the poster you have seen. She was the youngest scholar on biochemistry, PhD biochemistry she have awarded from Ramakrishna Mission Seva Pratishthan under the guidance of Dr. Mrs. Geeta Talukdar. She, and now Rinini is presently working as associate professor and senior biochemist in the same institute. She was graduated in chemistry from India, Kolkata University of Calcutta in Bethune College. Then she have done post-graduation from University of Kolkata and then PhD. Then, and she has worked on DNA analysis in forensic lab in UK. After that, she started in as a student in the Ramakrishna Mission Seva Pratishthan, and after completing that, she has joined in that same institute after a few years of experience in other places. So without wasting much time, I call Dr. Rinini to share her slides and talk. Thank you, Dr. Rinini. Welcome. You unmute yourself, please.
Just 1 second.
Yes, okay. We can hear.
Good evening, everybody. Thank you, Dr. Poddar, for inviting me.
Rinini, you just make full screen this slideshow in the PPT screen.
I have made it.
Okay.
Okay. You get it? I have made the full screen.
No, it is showing slide view. You click on the PPT view, the down icon.
Click first, enable editing, sir.
Enable editing.
Upside.
No, Rinini you have to do.
No, I have done it. I have done it
No,
for
no.
this slideshow.
At top of the screen, you can see enable editing.
Is protect view.
Yes, it's not there. It's not there. Enable editing, I'm not seeing.
Okay, you stop sharing, then you cannot see again. Wait, I stop sharing, then you can see. Then you start sharing.
Okay. Let me stop share.
I stopped. You check. Now you check the screen and make it full screen first, then start sharing. Because it is not enabled, the screen share, full screen.
So this is the full screen now. I have made it the. Is it now okay?
Now share screen, please.
Oh, share screen. So I cannot. Share screen.
Paul Chunku. Oh,
Share screen. Share screen and put it there. This one. This one. Make it full view. Make it full view.
Click first, enable anything.
Is it okay?
Ah,
Is
okay,
it
now okay. Yes.
okay? Okay. So let me start. Good evening, everybody. I feel extremely honored to be an invited speaker for the prestigious Professor Dr. Geeta Talukdar Memorial Lecture. For that, I thank Dr. Shondip Poddar, our beloved Shondip Da, and Professor Madhusnata De for providing me the opportunity to be present in this August event. Feeling a bit emotional this moment. So many memories flooded in my mind. Many years have elapsed, even after her demise, but I can vividly remember the first day, the rainy afternoon where I met her for the first time and expressed my desire to work under her. She was a renowned figure then, and she was a leading geneticist. She was a pioneer herself, an institution by herself in cytogenetics field and molecular diagnostics and various arena of clinical research. At that point of time, when I met her, she was doing work on hemoglobinopathies and thalassemias. So what I tell that her initial response to my request was not very much assertive, honestly speaking. So I got a bit disappointed, but all of a sudden she called me when I was about to leave, and she told me to come the next very day with all my credentials. My joy knew no bounds because I got the opportunity to work under her, such a stalwart in this field. It is needless to say, the next few years under her guidance was a smooth thing for me. I have learned so many things from her that I cannot count. It is countless. One thing I must share here on this platform, because what made her different from other guides, because she sent me to CCMB, Center for Cellular and Molecular Biology, a pioneer research institute in India. She sent me there. So this is very natural for a supervisor to send her student or his student to various places where they can learn. That is not big deal in that. But what made her different, because that time I was running out of fellowship, so she deposited money. At that point of time, it is 25,000. It was a big deal. So from her bank account, she deposited it to their account, to CCMB's account on behalf of me. And I was so moved by her, this kind of act, this kind of gesture, and I felt so much grateful to her because she wanted me to learn. That workshop was based on chromosomal and molecular diagnostics of various diseases. And when I came after learning from CCMB, I felt the pride on her face. That was a big thing for me. And the thing is that after getting my first job, when I repaid the whole amount to her, Didi, please take it. She took it. But she did not take it for herself. She donated it to the charitable organizations, the whole amount. That was her. She was so different. I'm so grateful to her, not only guiding me as a PhD guide, but I got unconditional love and support from her throughout my tenure. And after completing my PhD, whenever and wherever it is required, she was there for me. She left us in the year 2007. She's not with us. She's not with me. But I can tell the world that she resides in me. Maybe she will reside with me till my last breath. One thing I wish to share, that before coming to her, I joined the reputed institutes of Kolkata, Bush Institute and the Biophysics and Molecular Biology Department of Raja Bajar Science College. Both of them were pioneered still. But anyhow, due to some personal reasons, my PhD dream did not get completed. So she hold my hand and lead me in a righteous way to reach my destination. She helped me to fulfill my dream for PhD. So thank you, Didi. I used to call my PhD guide, not only me, all the scholars, they used to call Professor Dr. Geeta Talukdar as Didi. So thank you, Didi. Thank you for everything, wherever you are. You are, be in peace, and I know you must have watched me in these events. Because of you, I am here today. Thank you, Didi, for being with me in my life, wherever and whenever I needed it the most. So thank you very much, everybody, for giving me this opportunity. Thanks a lot. Now I will come to my topic, which is a very, very pertinent topic, that is role of vitamin D in autoimmunity, and with a special emphasis on Hashimoto thyroid disease. It is a autoimmune kind of disorders. So the thing is that vitamin D is a sunshine hormone. 90% of vitamin D is synthesized in our body internally, and 10% comes from food. So it is all well-known fact. Why it is called hormone? Because it has the structural resemblance with many steroid hormones, and it has the cholesterol as its precursor. Like hormones, this vitamin, fourth vitamin of the vitamin series, vitamin D, is involved in almost all biochemical and metabolic pathways, and thus it fulfills the criteria of being a hormone. So that's why we consider vitamin D not only the fat soluble nutrient, but also a hormone. So these are the known facts. I know most of the viewers know about vitamin D, but those who don't, a quick recapitulation for that. We know that exogenously we are obtained vitamin D by dairy products and fish oil, and endogenously it is synthesized on our body in presence of the UV rays. So 7-dehydrocholesterol, I will come to my next slide. It's there on our skin, and in presence of UV rays, especially UV B rays, it is getting synthesized. Vitamin D is renowned for its action for the absorption of calcium and maintaining bone mineral homeostasis. So calcitriol is the biologically active form of vitamin D. One thing before going to my next slides, so I should mention that I will concentrate on the vitamin D deficiency in the role of autoimmunity. So we should know that what are the deficiency level. So as per RDA 2012, it is when the serum concentration of vitamin D less than 20 ng per ml, the person is considered to be the vitamin D deficient. When it is 20 to 29, it is called insufficient. 30 to 100 is defined as sufficiency, and more than 100 ng per ml is considered the potential toxicity. So, but now here I'd like to mention, since 90% of the world's population are vitamin D deficient, so it is high time to reset the cutoff values of vitamin D deficiency. So there are various structures of vitamin D. These are big structures, cholesterol, 7-dehydrocholesterol. So you can see the big rings of the cholesterol moiety. Sources are very common for vitamin D. You can see from the pictures, the milk, the orange juices, the beef liver, cheese, egg yolks. Several supplements are there, cod liver oil, fatty fish, so many sources. You know we are taking every day, but still we are getting vitamin D deficient day by day. There might be another reasons, we'll discuss on that. So the activation, how it is getting activated. So it is there, 7-dehydrocholesterol is present on our skin, and argocalciferol that we are taking from dietary things. So both the argocalciferol and cholecalciferol or 7-dehydrocholesterol that is getting first hydroxylation into the liver in presence of the enzyme 25 hydroxylase. There, the first 25 hydroxy vitamin D is produced, and 25 hydroxy vitamin D is the determinator of the vitamin D status into our body. So 25 hydroxylase is the other name is the CYP27B. Second hydroxylation occurs in the kidney, and there the name of the enzyme responsible for double hydroxylation, the second hydroxylation, is that alpha 1 hydroxylase, that is CYP27B1. Then it is transformed into 125 dihydroxy vitamin D3, which is the active metabolite of vitamin D, and it has the tremendous power to exert its effects on various organs. So we are very much known about the endocrine functions of the vitamin D. As the picture shows that kidney, it is 125 dihydroxy vitamin D. It helps in the calcium homeostasis. Vitamin D is famous for its skeletal involvement. It helps in muscle health, bone health. It helps to regulate blood pressure. It helps to regulate cardiovascular health, neurodevelopment, etc. But it also exerts the paracrine and intercrine functions as well. So after hydroxylation in the liver, it traverses to the monocyte. There, the second hydroxylation occurs. That is the 125 hydroxy vitamin D monocytes, as you know, monocytes and macrophages. These then, so vitamin D receptors are there in the monocytes and macrophages, and it helps in the immunomodulation. So immunomodulation is a very important part of vitamin D, though many people are still not aware of that. So it helps in the prevention of autoimmune diseases, and as well it helps in the control of invading pathogens. The vitamin D receptors are also demonstrated on prostate gland, breast, colon, lung, and keratinocytes. And there, 125 dihydroxy vitamin D helps in this regulation of cell growth and differentiation. So we can say that cell growth and differentiation is regulated by also the 125 dihydroxy vitamin D, the metabolically active form of vitamin D, so it helps to regulate cancer as well. And many papers and some studies are there, the role of vitamin D deficiency in the development of malignancy. So metabolism, this is a repeat slide. Only the last portion, you have a look on the last portion. That is, after hydroxylation, that is 125 dihydroxy vitamin D is produced. Very fine. But you know, the body has its own mechanism so that we can get rid of potential toxicity by vitamin D. So in that cases, what happens, there are another enzyme that is CYP24 that causes the hydroxylation, 24-25 hydroxy vitamin D and 124-25 dihydroxy vitamin D, which is not so shown into slides. They are, there lies the inactivation. So the body itself helps to, so that we can get rid of the potential vitamin D toxicity. So just coming on the metabolically active form, it helps the intestinal absorption of calcium, it helps the bone resorption, it decreases the renal calcium and phosphate excretion. So now coming directly to the vitamin D deficiency, which is the hot topic in India, and it is a known topic as well. Vitamin D deficiency, mostly underrated clinical condition. So nobody gives any importance to the vitamin D deficiency and says it's only a deficiency of the nutrient, because they are unaware of the tremendous consequences of the vitamin D deficiency. I will discuss in my later slides. So most common medical conditions, it was first pioneered by Dr. Michael Holick. So he pioneered vitamin D research, and he told that the vitamin D deficiency should not be neglected and it should be given importance to delay the onset of many non-skeletal diseases. Autoimmune diseases is one of the non-skeletal diseases we will discuss. You know, 1 billion people. Now I think more than 1 billion people, 90% of world's population have vitamin D deficiency. It has been reported in everywhere of the world, nothing new in that, Canada, Norway, US, China, many other places. recent prevalence of vitamin D deficiency in India is 80 to 90%. So you can easily imagine that when you will go through the consequences of vitamin D deficiency, how it is important to look into the vitamin D deficiency. So we did a study in the West Bengal population. How much West Bengal population are vitamin D deficient? It was basically a population-based study. So we get in the year 2016, we did it, and we got 87.2% of the vitamin D deficiency was prevalent in West Bengal. There are various causes that causes the vitamin D deficiency. It might be the vitamin D's impaired availability, that is the secosteroid is not available. It might be the person might not get adequate exposure to sun. It might be the seasonal thing, because we know that seasonal variation of the serum concentration of vitamin D is very much demonstrated. In the winter, vitamin D concentration usually drops. It might be there are some conditions that is that the malabsorption of fat occurs. So if the malabsorption of fat occurs, then it ultimately leads to the vitamin D deficiency, just like celiac disease, Crohn's disease, etc. There might be the impairment of the hydroxylase enzyme, which causes the hydroxylation in the liver. So if the concentration of the vitamin 25 hydroxy vitamin D is decreasing, so naturally the precursor, if it is decreasing, so the next step will not occur. So there will be the deficiency. Similarly, if there is it might be the mutation into the enzyme that causes the hydroxylation in the kidney. So if there is an impaired hydroxylation in the kidney, so there will be the deficiency in the 125 hydroxy vitamin D. So there is a chances of causing vitamin D deficiency. Though it's rare, but there are some persons, especially the children, they are getting vitamin D resistant rickets. So then if you treat the children or the persons with the vitamin D supplementations, they get unresponsive to that. If a person's glucocorticoids on the prolonged time, so it inhibits intestinal vitamin D calcium absorption. I will discuss vitamin D polymorphisms as one of the potential causes of vitamin D deficiency. Why it is? Because there is a SNP, single nucleotide polymorphism in the vitamin D receptor gene. And if there is the mutations in the gene, that will ultimately cause the vitamin D deficiency, because despite availability of the vitamin D, no problem of vitamin D production. But the thing is that it is not getting transported due to the unavailability of the receptors. So these are the causes. So you have a look in this picture. So you see the consequences starting from the brain to leg. So we did it in 2017. We published a paper in the European Journal of Pharmaceutical and Medical Research. So there we focused on the role of vitamin D deficiency in the non-skeletal diseases. And you see there are all non-skeletal diseases, schizophrenia, depression, upper respiratory tract infections. So there are so many autoimmune diseases, type 1 diabetes. It is multiple sclerosis, it is rheumatoid arthritis. There is so much malabsorption diseases, Crohn's, people, cystic fibrosis. So which is not there. So many hepatic failure, renal diseases, so many diseases are there. So the thing is that what we studied and we published, you see, just have a look. I'm not going into details because it will take time. If you can have a look in the slide, you can easily understand that how much vitamin D the non-skeletal persons have. So all of them showed that less than 20, that is, they showed the severe insufficiency vitamin D. And eventually, these persons all have these diseases: hypertension, type 2 diabetes, PCOS, CVD, TB, CBA. So we did a study conducted on 2,700 vitamin D deficient people in West Bengal, and we got this. So now coming directly to the autoimmunity, concept of autoimmunity. So what is autoimmunity? When the system fails to discriminate the self and non-self, which is self-antigen and which is non-self antigen. When your body fails to discriminate these two, then autoimmunity develops and it mistakenly attacks its own self-antigens. So autoimmunity is autoimmunity is the failure of organism in recognizing its own constituent parts as non-self. They think they're self-antigens as the non-self antigens and elicit autoimmune response against its own cells and the dangerous consequences. There are multiple autoimmune causes that lead to autoimmunity. Preliminary, the genetic disposition is there, predisposition, that is the MHC2, major histocompatibility 2, MHC allele, there this DR antigen and DQ antigen. So these are mainly, so the genes coding DR and DQ antigens are mostly susceptible alleles for causes autoimmunity. And there are some these immune regulation and the environmental factors. Here I need to know, so you can have the genetic factors is HLA, DR, DQ8 and CTLA4, they are MHC genes. There are many genes others. Environmental factors, infection, stress, pollution, gluten, iodine intake, so they are environmental factors, and endogenous, so many factors. I will discuss only on vitamin D deficiency because of shortage of time. So the most important concept of autoimmunity is that if a person is genetically predisposed and if environmental exposure to that genetically predisposed person triggers autoimmunity. So it's not that any persons who are not genetically predisposed, there is a little chance, or I can say a lower chance of developing autoimmunity. But if the person is genetically predisposed, they are highly likely to develop this autoimmunity. That is the magic of the gene interplay with the environmental factors. So have a look of the autoimmune diseases, nothing much to say, so many autoimmune diseases. You can pick any organ. You can pick an organ, any organ of your body that can develop autoimmune diseases. So you never know which part of the body will elicit immune response of its own constituents. You never know. So now immune regulation. So as you all know from our childhood days, we know that there are two parts of immunity. One is innate immunity and one is acquired immunity. So any defect, a defect in any arm of this immune system can trigger autoimmunity. So acquired immunity or adaptive immunity. So there are two things. You know that cell-mediated immunity conferred by the T cells and humoral immunity that is elicited by the B cells. So now this is a slide. I will depict it in my next slide because that is a pictorial depiction. It will be easier for the audience to get it. So you just have a look on that. I have put vitamin D in the center. So it has the tremendous role on both arms of immunity. It is innate immunity and it is adaptive immunity. Both parts are there. What happens that let us first discuss the innate immunity. How it influences the innate immunity. You know that innate immunity is the first line defense of any immune deficiency. So it decreases the antimicrobial activity of the phagocytes. So you know the components of the innate immunity is that neutrophil and these phagocytes, monocytes. So it increases the antimicrobial activity of the differentiated monocytes and phagocytes. How? By secreting or by producing CAMP, cathelicidin antimicrobial peptide or defensin beta. So they secrete these chemicals and that destroy the pathogen. How it clears the pathogen? By chemotaxis or by phagocytosis. So I'm not going into details of the phagocytosis and chemotaxis because these are the very common and very, you know that, everybody knows that. So I'm not going into detail. Now what I tell that vitamin D also has the immune tolerance mechanism towards its self-antigen. That's why when the deficiency of the vitamin D occurs, it develops the autoimmunity because it promotes or it modulates the self-tolerance. How? So adaptive immunity, it decreases the pro-inflammatory cytokines, that is IL-6 and TNF-alpha. So it decreases the secretion of these pro-inflammatory cytokines by the T cells. What he did, vitamin D also increases the B cell apoptosis. So the antibody production will not happen because we know that humoral immunity also plays a part in autoimmunity development. So if there is a clonal selection, if B cells is getting multiplied, so there is a high chance of getting more antibodies. But if the B cells getting apoptotized, so in that case, its antibody production will be low. So that is a marvelous mechanism. contributed by vitamin D. What it does, it decreases the T cell cytotoxic activity by decreasing the fast L, that is the ligand, fast ligand expressions. It modulates the CD4 helper T cells differentiation by decreasing the Th1 and Th17. So these are the subset of the T helper cells. But let me tell you, these are the cytotoxic. So they will create the cytokines. And the other important thing, it induces the trait differentiation. That is the suppressor immune tolerance gene and increases the IL-tail and TGF beta. And most of the cases, say in case of the Hashimoto, I will discuss, it changes the Th1, that is cytotoxic phenotype, to the most immune modulating or immune tolerating system. What it does, it decreases the antigen presentation by monocytes and dendritic cells. So if the dendritic cells are not activated, so it will not present antigen. If the antigen is not present on the cell surface, so no question of T cell is getting activated. So what happens, that immune response will be hampered. So in that way, vitamin D plays a pivotal role in immune tolerance. So my next slide is that I have already discussed with the pictorial depiction. So coming to the autoimmune thyroid disease. So I have done some work. So it is basically the autoimmune thyroid disease. You see that is the Graves' disease and Hashimoto thyroiditis. One is hyperthyroidism and one is hypothyroidism. Hashimoto disease is a subclinical hypothyroidism. It is basically both of them T-cell mediated autoimmune disorder causing infiltration of the thyrocytes to the thyroid gland. Worldwide prevalence is 2 to 5%. But let me tell you that there is a very gender variation of the autoimmune diseases, and we are discussing the autoimmune thyroid disease. Yes, there is a extensive female preponderance that is occurring in the AITD. I have seen in the Hashimoto thyroiditis, the patients we took, more than 500 cases we did, and most of them, 90% were adult females. So there is some reasons because that skewed X chromosome inactivation might be the cause for the so much huge female preponderance of autoimmune diseases, because that is the only causes, and most of it, vitamin D also plays a very important role. So an interplay of genetic predisposition, I will discuss with a case report we got. So with these environmental factors trigger immunity. So now coming to the Hashimoto thyroiditis, it is basically a T-cell mediated disease and characterized by, you see, the goiter, presence of circulating anti-TPO antibody. Thyroid peroxidase antibody is there, and anti-thyroglobulin antibody is there. Anti-TG and anti-TPO antibody are the most prominent tests we are doing for the diagnosis of the Hashimoto thyroiditis. And we know that here also the intrathyroidal infiltration of B and T cells leads to the varying degrees of thyroid hypofunction. Close association of this HT with the VDD has been documented. So first documentations are there in 2009. So these are the symptoms of hypothyroidism. Nothing new in that. I think most of you know it. So if you don't know, have a look on that, the slide: hair loss, weight gain, increased fatigue, increased blood pressure, painful and prolonged menstruation, chills, convulsions. They are the common symptoms of hypothyroidism. So I have already discussed it. I am not going into details. The same thing, that is the existential factors, that the sex parity. So those who are having children are more likely to have this Hashimoto thyroiditis rather as compared to the nulliparous women. And genes are there. You can have a look on the genes: TG, TSH, thyroid receptor gene, thyroglobulin gene. These are the thyroid specific gene. HLA genes are the CTLA4, PTPN22, CD40. So these are the genes. And vitamin D, selenium, alcohol, smoking, stress, they are the environmental factors. Role of vitamin D autoimmunity, as I already discussed into my previous slides. So nothing new in that. I will just discuss in the Hashimoto thyroiditis is that vitamin D receptors are found in T and B lymphocytes. It is present in the macrophages. What happens? So if a person is the genetically predisposed, that means, as I told, that if a person is having the locus of DR or DQ locus in the HLA, DR, DQ locus in MHC2 gene, they are much likely to have this disease in audit when they are exposed to an environmental trigger. Now, one most important thing that most of people, they're not bothered about the genetic predisposition, because the genetic study is very difficult. It is expensive as well, and is a mass screening of the genetic predisposition is not possible in a country like India. I don't know the scenario in the outside, but in India it is very difficult. But what lies the importance of the genetic study? Because that HD and GD and T1DM and other autoimmune diseases, there is a sibling ratio. What does it mean? If a person having this disease, so I can explain with HT, Hashimoto thyroiditis, here the sibling ratio is greater than 10. You can imagine, or for Graves' disease, it is greater than 20. So if a person is having, if a child is having, or if a person is having this HT or GD, so it is 20 times or 10 times more likely their siblings to get afflicted with the diseases. So you can see, so if you can study, and if you can study whether the person is genetically predisposed, then the family members will be aware of these things, and they can avoid the environmental factors which triggered the autoimmunity. So here also polymorphisms of vitamin D I will discuss in my next slide. So what is that? Breakdown of immune tolerance after the interplay of the environmental triggers with a genetic predisposition. So there is a generation of larger number of T helper cells and cytotoxic lymphocytes. Then there is an accumulation of immune cells in the thyroid gland. And what happens? Prevalence of Th1 mediated diseases in the cytotoxic effects in the thyroid, and ultimately causes the apoptosis of the thyrocyte, and so hypothyroidism. So this is the pictorial representations, not going into detail for that. Now coming to my own work, what I did. So we did in 2015, because 2009 only this association of these HT and vitamin D deficiency first published in British Journal of Nutrition. In the consecutive years, cellular and molecular immunology, and there are very other papers, reputed journals, they depicted the association. But that these are all observational studies, and no such study was then there in 2015 in Eastern India. So we did that, and it was a pilot study. So sitting at the hospital, it's not possible to go the population study, but we got an indications. So in our study, we published in British Journal of Medicine and Medical Research. So we did the study on here. When it was published, it was 150, but later on we increased the numbers. So 78% of the vitamin D deficiency we got. 48.4. Can you imagine? That is the persons who come with the clinical, subclinical hypothyroidism and put on the ultroxin or the thyronorm or the thyroid lowering drugs. They are not knowing that they are having the anti-TPO positivity. And there is a significant correlation, negative correlation with the anti-TPO and vitamin D levels. So we just divided our study group in 3 cases. So I'm showing that this is a pie chart of this. So if you can have a look, that we measured the anti-TPO, vitamin D, PTH, calcium, phosphorus, ALK, thyroglobulin, every parameters, possible parameters which can upsurge the increase of the anti-TPO positivity. So we got that. So overt hypothyroidism based on TSH level greater than 10. So we just included some patients into the overt hypothyroidism category based on where the TSH level is greater than 10. When it is 5 to 10, greater than 5 but less than 10, that is subclinical, and when it is normal, that is the controlled population. You see there is a positive correlation of vitamin D with the anti-TPO in this control. But in subclinical hypothyroidism, there is a moderate decrease negative correlation. And in case of the overt hypothyroidism, there is a steep decline of anti-TPO and vitamin D. So now, the other thing, we also extended our study on the gestational hypothyroidism. So the pregnant patients who used to come, or you came for 2 years, we did the study, and we did the study for 365 pregnant mothers who came Ramakrishna Mission Seva Pratishthan for a routine checkup. So they have come for the routine checkup. So no question of anti-TPO testing was there. No question of vitamin D testing there, because let me tell you that vitamin D testing is an expensive testing. Anti-TPO testing also a very, very expensive testing. So in our charitable hospital, it causes 1,000 rupees, more than 1,000 rupees, or more than 3,000 rupees otherwise. So doctors cannot recommend that you go for vitamin D testing or you go for anti-TPO testing. The mothers, they come and they get routinely checked, and the other things for the pregnancy. But we know that vitamin D deficiency causes these gestational hypothyroidism, and most importantly, anti-TPO positivity. It poses a very detrimental effect on labor. It causes IUGR, that is intrauterine growth retardation. It causes preterm labor. It causes spontaneous abortion. It causes preeclampsia, so many things, mental retardation, so many things. So it does, but nobody is aware of that. These ladies, these women, they're completely unaware of their anti-TPO positivity, and the doctors, they were not. So whenever I approach the clinicians, please get them tested for anti-TPO positivity. So they told, how could they afford? But we, our group, need to spread the awareness that it is required. For that, we did on our project money. So what we did, we firstly screened 1,133 pregnant women. After initial screening, what we found that 47% of that GHT, and you know that GHT or gestational hypothyroidism also is a very detrimental or the very bad effect on pregnancy, but nobody knows of that. Though now TSH in the obstetric patients we get into biochemistry laboratory, but anti-TPO testing still no. 86.4% of the GHT patients having the vitamin D deficiency, but they are not tested. They are tested for our own interest, for research interest, but they were not recommended to go for vitamin D deficiency in the year 2015 to 2017. But I must say that after publishing these studies and after going to the clinicians and spreading awareness and after conversing with the clinicians, now the testing of vitamin D deficiency has increased definitely. 72.3 euthyroid pregnant patients who didn't show the elevated TSH level, they were also vitamin D deficient. 28.7, can you believe that 28.7% autoimmune thyroiditis was observed in the GHT patients? They were completely aware of that when they came and asked, what is that? So we just asked them, we advised the patients to go and show this report to the clinicians, so they will do the needful things. To our surprise, that is our study also demonstrated 3% autoimmunity in the euthyroid women as well. But it is documented. So because I have seen many papers who documented or demonstrated that 1 to 2% of autoimmunity in the pregnant women. So naturally, so it was our study. So there are many studies there. That time we are working on autoimmunity. So we renowned endocrinologist, he collaborated with us and he gave us 51 type 1 diabetes patients. And we tested GAD, glutamate acid decarboxylase enzyme for that. And you know that we got 36% of the T1DM patients who showed the GAD positivity. And you know, the persons who are GAD positive, they are most likely to develop Hashimoto thyroiditis or autoimmune syndrome, that is polyglandular endocrinopathies. So you know that So our study in this type 1 diabetes, on type 1 diabetes patients, it was selected for poster presentations in International Diabetic Federation there. So that time getting 50 diabetic patients in India, in West Bengal, is a big task, and we did that, and we see that is the yes, vitamin D deficiency was there. And most of the type 1 diabetes patients, I must say that 36%, so not all 36%, but if I can remember correctly, 15 to 20% of the T1 DM patients, they showed HT, anti-TPO positivity. So now coming to the interesting case, that is the polyglandular autoimmunity with Hashimoto thyroiditis. So we got a case. A girl of 6 years, she came to our lab and with all symptoms of diabetes. And so her sugar level was very high. If I can recall correctly, her FBS was, it's very high, 373, and her PPBS was 500. And her C-peptide was very low, so it satisfied all the criteria for type 1 diabetes. She was admitted under an endocrinologist at our hospital. After 1 week, she developed some symptoms of the swelling, and we tested anti-TPO, and anti-TPO level was some 1,000. Then we did that. These patients showed the type 1 diabetes. This patient showed the HT, TSH level was high, and then we did the other tests. We did also for vitamin B12, we did cortisol, and we did that the other test to identify or to categorize in which category this girl falls. So it is so we ultimately categorized this girl into the polyglandular autoimmune, PAS 3A, because the girl did not develop in alopecia, or it does not develop in Addison's disease, or it does not pernicious anemia. She didn't display the symptoms, or because her vitamin B12 level was within the normal range. So in that cases, we can say we categorize. So, and when we got this, then it intrigues us the whether this girl is genetically predisposed. Let us see. So we got the antigen testing, genetic testing, and this girl shows HLA-DR and DQ locus in her. So, and upon investigation, it revealed that this girl was on cow milk. So what we can see is that upon consumption of the cow milk, so a single case cannot determine the genetic interactions. But the thing is that it is an indication, a case report, but it is an indication that satisfies that gene environment interactions plays an important role in developing autoimmunity. There are so many polymorphisms and VDR polymorphisms are there with AITD. Just excuse me, I need to switch on my phone. Thank you for your patience. So we did polymorphisms with AITD. So we got that if there is a single nucleotide polymorphisms in the vitamin D receptors, that is the VDR polymorphism. So FOC1 polymorphism is very much associated with the HT, type 1 diabetes. It might be there because we know that MHC gene, it causes the overlapping regions of many autoimmune diseases. That's why it clusters in a family. So if one autoimmunity is there, so there are many papers in type 1 diabetes paper in South India, and outside India, there are also papers, they demonstrated the association. So this might be the cause because the genetic overlapping regions of many autoimmune diseases. So that leads to the production of this autoimmunity. So FOC1 is that it's the exon 2. There is a polymorphism, consecutive 2 codons, there is a mutations, and there is a T2G, if I can remember. So that is important for causing autoimmune disease. So vitamin D polymorphism nobody studies. So my question is that if we are having milk, being an affluent family, if we are having fish, if we are having many things, the sources of vitamin D, it is true that we are not exposed to sunlight, adequate sunlight so much, but we did many things for getting our vitamin D adequate. So vitamin D polymorphisms might be considered for causing vitamin D deficiency in today's arena. Now coming to the most pertinent point, that is the effect of vitamin D supplementations. It is a very, very recent study. It is 2018 study, I suppose. They did a randomized double-blind placebo-controlled trial. There, the effects of vitamin D supplementation on the thyroid function in hypothyroid patients, and they showed that after vitamin D supplementation, so definitely thyroid function getting improved, anti-TPO level getting down, TSH getting down. I can share a personal experience of mine. So my daughter, she also developed anti-TPO positivity when she was very stressed in her 12th exam. But after admitting in the engineering college and after coming to this, so then she was supplemented vitamin D, and without, believe me, without putting on Thyronome or other thyroid lowering diseases, she's now perfectly okay. So one case might not be conclusive. So I have not done vitamin D supplementation on the hypothyroid patients because doctors are not agreed, because there is also a controversy whether the supplementation improves the thyroid functions, but I'm really interested to do that. So it opens a new avenue to treat non-skeletal diseases and archives of disease in childhood. These papers demonstrated a meta-analysis of these observational studies, and they showed that type 1 diabetes is significantly reduced in those who are supplemented with vitamin D. And they also told that incidence rate of type 2 diabetes is 50% lower if they are given or they're supplemented with the 25-hydroxy vitamin D. So it has been demonstrated in many studies that it reduces the incidence of diabetes, PCOS, CKD, COPD, and cancer with vitamin D supplementations. So there are in 2016, that is the Food Safety and Standard Authority of India, they did a study and they said that standards for fortification of food items like salt, edible oil, milk, wheat, and rice with iron, folic acid, vitamin D, and vitamin A might still doubt whether the patients are getting fortified vitamin D products. So from around 60% vitamin D deficiency in the beginning of the century, it has now come down. It is an old slide, 2013. I think that it is now getting importance among the clinicians, so that vitamin D testing has been increased. In my day-to-day lab practice, I have seen, and definitely they're working on that. So conclusion is that vitamin D deficiency should not be ignored. It should be considered as a public health problem, though it is underrated, but we should not ignore it. So it should be recognized by the government body so that the healthcare funds can be allocated appropriately, and implementation of population-based educational and interventional strategies to combat this serious problem. Otherwise, what we have done, we have this the hospital-based study. So large participants population-based study is very, very essential to reach a conclusion. So I'm deeply acknowledge the Ramakrishna Mission Shiva Pratishthan and the secretary and my students and my laboratory technicians. They are really, really very supportive. Whenever I require any help, my technicians, they extended their help to any extent. They never said, madam, we cannot do it. Whenever, they are not paid for that, but they helped me to draw blood, to do tests, everything. So I really acknowledge my laboratory medical technologists. And definitely I acknowledge Professor Geeta Talukdar, because she instilled research instinct in me. Though I'm working in a biochemist, I'm not in a proper research lab. So, but I am very much eager to do research, to do more things for the betterment of the mankind. So I thank you everybody for your patient hearing, and I also thank Michael Holick for pioneering the vitamin D research. Thank you very much for your patient hearing.
Thank you, Dr. Rinini, for your nice presentation. And I would like to ask everybody to raise their questions, if any. I also put the query in the YouTube. So still YouTube, there is no question. And if in Zoom you want any, you have any question, please. Anybody can unmute themselves and question. Otherwise, we hand over the microphone to Prof. Guswarani.
Okay. So thank you, Dr. Sandip, and also Dr. Rinini for the really good presentation. So even though my field is different from you because I'm from forestry, but it's really good to know about, I think this vitamin D is really important for us. I mean, yeah, I just is surprised this your your research result about, if I'm not mistaken, about 87.2% for vitamin D deficiency
More
in
than that, I think.
yes. So
Yes,
I
it's
think
more than that.
maybe also happen in Indonesia as well, because about this, this problem in the world, like you mentioned before.
Yes.
Yeah, sorry if I give a question first. I think this everyone from medical school here, from the Faculty of Medicine, and also from I think biology, chemistry. I think this maybe I think this is your turn to ask Dr. Rinini, but it's okay because Dr. Sandip already gave me a chance for the first time. Thank you. This is my question. Just I'm wondering to because you mentioned about for the vitamin D, this for the cause of deficiency because of the hereditary. Yeah. So from the parents, they inherit to their kids or the children for the vitamin D deficiency. So
Not to vitamin, pardon, madam, it is autoimmune diseases. So autoimmune diseases for that.
Okay. So
Yes,
not
yes.
just for the vitamin D. So that's why I'm just curious about that. Yeah. Also, I think for the autoimmune, also this is most common problem also in Indonesia as well. I have some friends. They have problem also for their health about the autoimmune. Yeah, I think the term of autoimmune is like, we just know this recently. In few years ago, yeah, especially for me, I didn't know about this autoimmune, but I know some friends they have a problem about that. So this I'm curious about how, yeah, I mean this specific cause to to make this autoimmune for the people, because for example, I have a friends, a woman, and yeah, she just got autoimmune after she delivered her kids for the second one, and for the first kid is okay for her. Yeah, she was very healthy, but after delivering her baby, and she got autoimmune, and until now. So maybe a little bit you can also like share us how what's the problem to create also
It's
to
not
make.
like that. It might be that time she was diagnosed, it was suppressed, that she might be genetically predisposed, or it might be that in that time environmental factors, she being exposed, that triggers autoimmunity in that point of time. You never know when the environmental triggers to develop autoimmunity. So you never know. So this time I'm okay, but I know that in coming years whether I will develop autoimmunity. So that's why it's very important, the environmental factors which causes autoimmunity. So we need to check on the environmental factors, and similarly we need to go for the genetic testing. Though it is not very feasible, but if we can opt that, then we can have a restrict on that, that we should not have these. See, I'm telling you that if as I discussed the child, so in my case report, if it is well known that she is genetically predisposed, and if it is well known that for type 1 diabetes cow milk is an environmental factor, so in that cases, if she know that she is genetically predisposed, so she should avoid the environmental factor that triggers the autoimmune response. So in that way we can restrict our exposure to the environmental causal agents that causes autoimmunity. So that's the thing. Whenever it occurs, nobody knows.
Okay, thank you. So maybe for the second question, Dr. Sandeep, yeah, I'm just curious about between environmental factor and also for genetic factors, which one is the most serious problem to make for the autoimmune? Because right now we live in a lot of pollution, and yeah, I think, yeah, I don't know, like from the scientific journal or from based on your result, which one, genetic factors or environmental factors?
Madam, it is a genetic and environmental interplay. It is it is an interplay. Not only that, everybody that I am having cow milk or I am having something which causes autoimmunity, but I'm not genetically predisposed. So might not that environmental factor will lead to cause autoimmunity in me. But if I am genetically predisposed, might be that environmental factor causes or augments or enhances to develop autoimmunity in me. So that might be a balance of that. When your T cell will upside, so the vitamin D, I think it is why I'm emphasizing that, because nobody knew about that. Everybody knows that vitamin D is important for skeletal development, for bone mineral hemostasis. Nobody knows that it is so important in non-skeletal diseases. So if having supplemented with the vitamin D or these things which can increase our serum concentrations of vitamin D, so in that cases we might delay the onset of the metabolic diseases. We might delay the causal agents for the autoimmunity. So in that way we can do it.
Okay, thank you so much for this really good explanation. Yeah, thank you.
Actually, madam, it is true, and vitamin D have many effects actually, and as many of us not exposed to sunlight,
yes,
we cover our body with every time we are in office inside the dark room. So that sunlight not producing means touching our skin, and so we all of us getting vitamin D deficient.
Yes.
And now the milk also there, and milk is we are going for fat-free milk. Actually, fat-free milk is without any chemical. That is not milk.
Yeah.
We don't know, but we are health conscious. We go to shop, we search for fat-free milk.
Yes.
So that is not giving the source of calcium and vitamin D at all. So these are many factors are there, and these environmental factor, who is exposed and whom it will trigger, that is that is controlled by our gene.
Okay.
So somebody is getting exposed, and as Dr. Rinini told. So, madam, thank you, madam, for raising good questions.
Thank you so much.
Yeah, okay. And another question is there from Dr. Seema Singh. Yes, Seema, you want to speak, or I read the question? You can unmute yourself and ask.
Hello.
Yes, Seema.
Hello. Good evening to everyone, sir.
Yes.
Sir, my question is that for vitamin D deficiency, only the sunlight and vitamin D medicines are the solutions or anything other? Because one of the member in my family is suffering from the kidney related problem, and also he is deficient in vitamin D. For that kind of person, what we should take precaution or anything which is good for recovering in case of vitamin D actually?
Yes, Rinini, you want?
Yes.
Yes.
I want to answer. See, in case of the CKD or nephrotic syndromes, as you mentioned, that we get patients for that who are vitamin D deficient. So exposure to sunlight for getting covered by vitamin D deficiency is not feasible. So what doctors, they recommend the vitamin D supplements. So I think vitamin D supplements are the way to replenish your vitamin D deficiency. And not only that, that after there is a dose you can cover to recover vitamin D deficiency, and there will be a maintenance dose you need to follow to be vitamin D sufficient or to maintain your vitamin D level. So supplementation is the way doctors usually supplement the vitamin D. So for myself, I can say that I was severely vitamin D deficient. It was less than 8. So our autoanalyzer CLIA platform, they cannot measure vitamin D below than that. I was that much sufficient, but I was totally asymptomatic. So I was put on supplementations. I took the supplementations and I'm okay. So I think supplementation is the answer, and that way you can get rid of vitamin D deficiency. Or the fortified food, that can also help. But exposure to sunlight, I think it's not practically feasible, or that can improve your vitamin D level to that extent.
Okay,
Thank
ma'am.
you.
Thank you so much. Thank you, ma'am. Thank you,
Thank you,
sir.
Shima. I would like to introduce Dr. Shima is one of our ex-student, and she already awarded PhD, and she is now government college lecturer in
Thank you, sir.
India. Thank you, Shima, for joining. And
Thank
another
you.
thing must tell that as he is suffering for kidney disease,
Yes,
maybe
sir.
it is for he has uric acid or gout problem. So
Yes,
we
sir.
must check that one, because if we take more animal food, animal source protein for vitamin D replacement, so maybe it will increase the gout chance, and that will increase the kidney stone and etc. formation. So if uric acid is there, very dangerous. So we must control that. Otherwise, kidney dialysis and kidney failure may happen. So that is very important aspect of what you call the gout, uric acid. And as Dr. Rinini told, supplement of vitamin D, as all of us know that vitamin D is the family member of ADEK, fat soluble vitamin.
Yes,
So
sir.
if it is hypervitaminosis, then it
Hmm.
will not go out from our body easily. So we must take a controlled dose, and we must take proper, what you call, with proper monitoring. Not take simply vitamin D, somebody taking, so I also take, not like that. So we should consult with doctor and test, and accordingly we should take. Vitamin D we should not take abruptly, as not only vitamin D, all multivitamin, ADEK.
Yes,
Vitamin
sir.
B complex, no problem. We can take, it will water soluble, it will go out with our urine. But if vitamin D or fat soluble vitamin, that is
Yes, sir.
difficult. So we must suggest also that, as we told, that cod liver oil and that type of sea fish is the sources of vitamin D, and milk. So we can go for that. So that is
Okay.
another option. So
Okay,
any
sir.
other
Thank you.
questions from any? Thank you, thank you. Any other questions? Any student? I think no question more. So,
Maybe we can
Madam,
invite also from Indonesia, maybe some.
yes,
Yeah, we have.
Miss Ulifiti is there, one of our students
I have
from
question,
Keynote.
Yeah,
Prof.
Dr.
Yes,
Agustin
yes,
here. Dr.
Dr.
Agustin.
Agustin.
Yeah, go ahead.
Yes, yes, Dr. Agustin, please.
Thank you very much. It's also interesting for us because every day we are talking about the pandemic COVID-19. So, do you think it is also have a benefit for consume vitamin D for our body for treatment of COVID-19? The first, and the second one, if in our body have too much vitamin D, is it dangerous or no? And how can we analyze with the easy method, maybe, if our body is need or sufficient of the vitamin D? Is I think it's all of my question. Thank you.
Thank you, Dr. Rini. Hello, Rini. Hello,
I think
Dr. D.
problem with the connection, maybe. I
Oh.
think. Yeah.
Yes, if I can give some, means if it is vitamin D excess, it will have some problem. And hypervitaminosis of vitamin D, everybody can see in the book. And if it is means increased amount, as I told, it is not water soluble, so it will not go out with urine so quickly. It will stay in our body, so it will cause some problem if it is hypervitaminosis dose. So we cannot take, suppose the dose is 1 tablet for, means 1 capsule per day, we cannot take 10 capsule like that. But in case of vitamin C, in case of COVID, we are telling that you take vitamin C more. It will boost your, so vitamin C, if we can take 4 tablet, means 4 into 500 milligram, it is around 2 gram per day. That also will not affect bad effect. But if we take vitamin D in that respect, to a 2 tablet or 4 tablet, we will feel problem. Okay. So this is one answer. And about the COVID relationship, I am not aware about that because I have not so much experience in vitamin D. So I am also aware.
Yeah, because there are some treatment if we exposure with the sunlight in the mornings, is also increase our antibody as well.
Yes,
And
yes.
that we see is also important for COVID-19 treatment as well. So maybe it's also have the theory about the vitamin D in here.
Just আবার কানেক্ট করো জুমে okay please come. So she is joining, already lost connectivity.
I see.
Actually, there was a cyclone before 1 month, as madam knows, in Kolkata, and after that still line is not recovered everywhere, internet.
Okay.
Like Professor Mulsade also cannot join today, very poor, she cannot talk in WhatsApp also. And also now few days very raining now again.
Okay.
So that is the problem now. So she is joining.
Okay.
And actually other cause of vitamin D, we know that in Malaysia and other places here, actually many persons suffering with diabetes. And that diabetes actually causing the osteoporosis.
Yes.
And this is not only diabetes causing osteoporosis, vitamin D deficiency also there. Because we cover our body. That is one of the reasons. So sunlight cannot touch our body. We stay in our AC room. We don't go to sunlight. If we do walk, we do evening walk. We don't do morning walk because morning walk sunlight can come, but we don't do morning walk because for the heat, means total heat sun here in Malaysia. If we go to sun, we will get sunburn. If we stay,
I'm sorry, my net got disconnected.
yes, the question is for you, Dr. Rinini, that Dr. Agustin asking that is there any relationship of vitamin D and COVID we can think?
Yes, you can. I can say,
Yes,
though we have not studied, but since vitamin D elicits immune response and it has a very protective role of immunity, so we know that in the immune suppression occurs in COVID. So there is so definitely there might be a relationship of the vitamin D with the COVID. I suppose, though I have not studied or I have not got any studies, but there should be a relationship. See, vitamin D causes the immunomodulation, so there might be a role. That might be a relationship of vitamin D with the COVID.
And another question he asked that what is the hypervitaminosis activity of vitamin D? If we take
Hypervitaminosis, yes, if it is greater than 100, so it is the potential toxicity. So it's getting deposited into liver, kidney, and other organs. So there are some hypervitaminosis activity. The fatigue, cramp, muscle cramp occurs, fatigue occurs. So there are some symptoms that leads to hypervitaminosis D. So it is very dangerous. So if we get greater than 100, so we usually report to clinicians to get the necessary measurements for that. So there are some symptoms of vitamin D.
So how can we analyze ourselves to know is the enough or not in our body? Is
Yes,
it some instrument for analyze that?
Yes.
yes, yes. Let me tell you. That is vitamin D. There is a test. It is a diagnostic test. There we are measuring in an autoanalyzer. That is the methodology used for vitamin D. It is the chemiluminescence. So enhanced chemiluminescence method we use, and there are some autoanalyzers in biochemistry where we measure or we test vitamin D, and there is the reference range of that. If your serum concentrations of vitamin D, usually we measure 25 hydroxyvitamin D total. That means that the endogenously synthesized onto your body and what you are taking from dietary sources. So the total 25 OH vitamin D we are measuring, and if your serum concentrations is less than the deficient range, that is less than 20, then you are deficient. If it is 20 to 29, then it is insufficient. If it is 30 to 100, then you are sufficient. And if it is greater than 100, then you are toxic. So in that way, you can get yourself tested for vitamin D.
Thank you, Dr. Rinini.
Thank you.
Actually, I worked in lab also, diagnostic lab, but I worked in molecular biology aspect of diagnosis of HIV, etc. in diagnostics lab. But I see the chemiluminescence machine. I know that the range, etc. She is the best person because she handling that part
Yes,
in the lab.
we
She
are
is
dealing
managing
with
the whole lab,
autoanalyzers.
biochemistry lab in the hospital. So thank you, Rini, for your answer. And
Thank you.
So,
also, I would like to say that I worked on iron. Same thing. If we give ferrous sulfate as iron supplement for the pregnant women. But if the ferrous sulfate, it is more we take, it is poison. It is great poison, and it is named as Hirakosh in Bengali. So it is a very toxic thing.
Iron toxicity is also
So,
there.
and that will lead to hepatomegaly and other diseases. So we will discuss that one on another meeting, madam.
okay.
And thank you for
Thank
everybody
you.
joining.
Thank you, madam.
And
So another question, Dr. Sandip, please.
yes, we give the token of appreciation
So I
certificate.
just read from on YouTube.
Yes,
Okay.
I show the certificate. You can see.
Yeah.
Thank you. This certificate offered to Dr.
Thank
Rinini
you. Thank
Dastidar
you so much,
Question.
Dr.
for
Poddar and
certificate
madam.
appreciation from all of us. From
Thank you
my
very much.
father, Dr. Suryapada Poddar, President of Dr. Tarak Poddar Memorial Foundation. My brother is around in this chat. He is also Professor of Ayurvedic Medical Science. And Dr. Alan Puri, the Rector, Patpetula University. I think sir is not around. Madam Gushwaran is around. He is the Vice Rector of Patpetula University also. And she has another post in Bengaluru University.
Yeah.
And Dr. Awal, Dean of Faculty of Medicine Health Science, and Professor Dr. Irfan Gastian, he also around in the chat box, I see in the participant. So from all of us, we thank Dr. Rinini and also for participating, and all participants will get the certificate because they have filled the response form, and it will take maybe 7 days time. So you will get by auto, because per day it will go around 100, etc. So, and also I am thankful to all senior members. I have seen Dr. Mala Bush and Dr. Ajanta Haldar. Many senior persons are present in this meeting room. Thank you all for joining. And also, madam, thank you for joining the medical
Dr. Sandip,
faculty.
excuse me, Dr. Sandip.
Yes.
Do we have time for, I get the question from here from YouTube.
Oh, okay. Yes, please. Yes.
Yeah. So sorry about that, Dr. Rinini.
No. Yes.
This is from Dr. Asmi, medical doctor. Right now, he is as Vice Rector for Student Affairs at Faculty of Medicine and Health Science at University of Bengkulu. His question first, he has two questions. Question number one, what is the signaling mechanism that is affected by vitamin D in this autoimmune disease? FOC1 or something else?
So
And
yes.
yeah, also, yeah, I think, I can continue for the question number 2. Will vitamin D fortification in children reduce the risk of autoimmune disease in the future?
Okay.
Is it okay?
So
Yeah.
yes,
Very
yes,
good
I can
questions.
hear, madam.
Yeah.
The easy one, I can say the fortification of food for the children to get rid of vitamin D deficiency. The research is also going on. So there are many controversial output. That is, some group of researchers, they found that supplementing vitamin D or fortified food that improves the vitamin D level. A group of researchers says, as I told you, that in archives of child disease, so these paper, they demonstrated, there are other papers also, they demonstrated that upon fortification of food, if we can take, if the children are put on the fortified vitamin D food, their vitamin D levels increases. But a group of researchers, they said that it's not. So it depends on your locality or your country, and the observational study I recommend, that whether it will help. So you can go for fortification and you get it tested, the children, that whether these level before fortification and after fortification, so whether it is getting improved. If it is improved, so you can make a pilot study on that, and then you can extend the larger participants, and you can have a conclusion. So that is, I recommend, that is, you have to go for your own study, but there are many studies, both of them, that a group of researchers, that is the conflicting answers are there. Fortification of food. And your first question is that I have already explained in my slides that there is an intricate mechanisms how vitamin D immunomodulate. The immunomodulatory role of the vitamin D is very much appreciated nowadays. That is how it decreases the pro-inflammatory cytokines. So it protects your self-antigen by the B cell or T cells, B cell humoral response, B cells elicits humoral response. So vitamin D causes the apoptosis of the B cells. So the vitamin B cells cannot further go for clonal selection. So it cannot multiply. If it is cannot multiply, then what will happen? It will not secrete the autoantibody against the self antigen. And similarly, it decreases the cytotoxic T cells activity. So most of the cases, what happens, that we know that T cells are two types. That is, the helper cells are two types, that is CD4 and CD8. So what it happens, that it decreases the antigen presenting cells activity. If the cells, dendritic cells, phagocytes, monocytes, if they are at the cells, they present the antigen on the cell surface, and it invites the T cell to attack that. There is a mechanism that vitamin D, it decreases the concentrations of the antigen presentation. And if the antigen is not presented, then T cells will be of no use. So it will not elicit immune response. So in that way, vitamin D helps in the immune modulation, or you can say that immune tolerance mechanism.
Okay, thank you, Dr. Rinini. And also thank you, Dr. Ahmad Asmi, for this your question. Dr. Sandip, we have also another question on the room chat. You can see from Dr. Sima Singh, I think.
There is one, I think, in Malay, in YouTube. Jam Barapu Wakta Tepa Tantu Mindakan Sinar Matahari Kirim Sinar Matahari juga Minang UV yang juga bahaya untuk kulit. Madam, I copy this.
Okay, so I didn't see here. I just
from Irma.
Oh, Irma.
No, Irma Badriana.
Irma Badarina. Okay, this is from University of Bengkulu.
Yes.
Okay.
This is the question I write here in the chat box. Zoom.
Okay, let me see first. Maybe you can also answer first. There's another question from Dr. Sima Singh before.
No, already done. I think Sima Singh asked.
This is about the deficiency vitamin D medicine.
Yes. Already done, madam.
Okay. Let me see for the YouTube first. Okay.
I give in the Zoom chat box, madam. That Malay one.
Irma, okay. This her question from Irma Badarina. what time for the good time to get this for the sunlight? Yeah.
Sunlight.
Because the sunlight is also this related to the UV and also for dangers for our skin. So that's why she asked about what specific time this good time for us to get a touching for the sunlight. Yeah, thank you.
The answer I can say that is the exposure of the UV or B light is essential for synthesis of the vitamin D on your skin. So 7-dehydrocholesterol, that is getting vitamin D in presence of UV or B light. Now it depends the zenith angle of the sun. So zenith angle is an angle how it radiates. So if the zenith angle of West Bengal is different, might be different from Malaysia or from other parts of the India. So depending on the zenith angle of this sun, the exposure we get, that depends. So it might be somewhere else that 8 to 10 is a good time. Somewhere it might be in the peak noon, in the 12. So it depends on the zenith angle, more or less when you get the adequate sun exposure. So I think that is it is 10 to 12. It is the mid noon. So then you can expose, but it is a very difficult. Some doctors say or some researchers says that you should expose yourself into the morning sun. Okay. So it depends on the angle. The important thing, the concept of that, you need to get the UV and B rays. So that is essential for synthesis of your vitamin D. So depending on that, you can say. And the morning sunlight is not when the sun gets up, not very much that is you don't need to be scorching sunlight, but you get adequate exposure of the sunlight. That will be sufficient to synthesize your vitamin D on skin.
Yes, you are right. Actually, in 10 a.m. in morning, if we go out in Malaysia, it is very hot.
Yeah.
Yes.
And if it is morning 7 a.m., it is dark, no sun yet. So in Indonesia, maybe 6 a.m. it is dark, and 9 a.m. is very hot.
Yeah, exactly.
We cannot go out. So within
But
7 to 8
I'd like to share that in the rural case also, where the farmers and all, they are working in the field. So they're getting exposed into the mid sunlight. So they should not get the vitamin D deficiency, but it happens. So I think that there might be vitamin D receptor polymorphism that might need to be investigated, whether there are hidden causes of vitamin D. As he also pointed out that we are very health conscious. We are applying sunscreen while going out in the sun, so that to get rid of, we shouldn't get darkened. So
no,
that
there is another thing also there. In here, they don't go out in field farmers. They cover their body when they go for work.
And that is also a cause.
So,
that might be a cause.
Okay, thank you, Dr.
Okay.
Rinini.
Other question, Ms. Yoga also answered already. Same question. So thank you, madam, for checking. My YouTube was hanged, so I cannot get the question.
No problems. Yeah, okay, my pleasure. Okay.
Thank you all for
Thank
joining.
you very much.
Thank
you very much.
And next week,
Nice to
madam,
meet you
we are
all.
going to do the 1 week program from next week. I am making the schedule. I will give to everybody so that everybody can join.
Thank you.
Okay.
Thank you, Dr. Podda, Shondipda. Thank you very much.
Thank you for joining.
Okay, madam.
Thank
Thank
you,
you
madam.
very much for your interest.
Thank you all
Thank you.
for joining.
Thank you very much.
Okay.
Thank you.
Thank you.
Thank you, sir.
Yeah, thank you, Professor Irfan, the Dean of the Faculty
Yes,
of
sir.
Mathematics and Natural Science.
We cannot see you, sir.
Yes.
We can
Maybe
now.
Professor Irfan, you want to say something?
Yes, please.
Yes. Okay. Maybe Dr. Sandip, we can invite Professor Irfan.
Yes. Prof?
Yeah.
Okay, Dr. Sandip.
Yes, sir.
Thank you for your invited our
for
faculty
joining.
to join this forum. I think the next time we will be join like this program. In our faculty, we have nursing program study and white
Free, yes.
white program study. I think it's good for me. Thank you.
Yes. Actually, in Lincoln, we have nursing diploma to PhD, diploma, bachelor, masters, and PhD in nursing.
Okay.
Medical also have, dental also have,
Yeah.
also pharmacy diploma, my masters, bachelor, masters, PhD,
Okay.
full
Okay.
level, and also management. So agriculture also now starting bachelor of agriculture. So biotechnology also there, masters and bachelor level. And PhD applied science is there, PhD nursing, PhD health science, PhD medical physiology, many programs are there. So we are also doing many conferences and also journal publications, etc. We'll
Okay.
definitely
Thank you.
collaborate and invite all of you to what you call encourage the students to take part of all these events so that everybody can aware and get some information.
Okay.
Thank you, Dr. Irfan.
Thank you.
Thank you
Yeah.
for coming.
We are very sorry, Dr. Sandip and also Dr. Rinini, because Dr. Ahmad Asmi, he couldn't join us because the problem for the connection, but he's watching and join us on YouTube. Yeah,
Oh.
so maybe he cannot.
Thank you, sir, for joining.
His speech here. Okay.
Thank
And
you.
also, Professor Alnopri also sends the warmest regards. To
Yes.
everyone here because he right now he's traveling from Curup to Bengkulu. He just sent message to me. He's very sorry cannot join us, but just enjoy for the lecture. Okay, thank you.
Thank you. Thank you again. And I must tell again, I remember Dr. Geeta Talukdar as in memory of that. And she always encouraged all women scientists.
Yes.
As you know that she was the scientific advisory committee member of government of India, central government, and she is the in charge of Eastern region of women development. All women she encouraged. Like Rinini, many women researchers are there working under her and get graduation.
She encouraged a
And
lot.
she encouraged, and as you see that Dr. Madhusudhan Dey, she left the government job for working as a scientist, research position under her. So that is the thing, and she is also very helpful. Even she suffered from cancer in the final stage. From cancer bed, she knows that what his scholar can do, her scholar can do.
Yes.
She told me that you join there, but you will leave after 1 month, I know. Because what I will do after 1 month of one service, she know already that this person will not work there for more than 1 hour or month. But she recommended me for go there like that. So
Shondita, I would like to share that is that time she was very ill. Didi was very ill, and then there was an international symposium on thalassemia and hemoglobinopathy.
Yes, we
and
both are there. And
director,
that
we
told
both were there.
that the director
so
general
feeble,
was
she
there
was not
from
able to
government
work.
of India. Why you not ask for job?
Yes.
But she knows that we will not ask. So that type of inner thoughts she has inside. So thank you for joining with us.
Thank you. It's a privilege for me.
Thank you.
Thank you, madam.
Thank you.
Thank you. Thank
Thank
you,
you all of
Rinini.
us. Thank you.
Yeah. Okay.
So we end the meeting, madam, with your permission, sir. Dr. Irfan, we end the meeting. Thank you.
Leaving meeting.
Okay. Thank you so much. See you. Bye-bye. Okay.
Bye.
Stay safe. Stay well.